Heck yeah. Well, if we're recording, let's start. And definitely want to start by saying hello to everybody. Thank everybody so much for coming. These Sunday events have been amazing, all thanks to Jerry for organizing. And you know, I think last week with Bosch's incredible presentation on fine-tuning stable diffusion, we've really reached a new level of production and professionalism. And I'm so excited about this being a place where, you know, on Sunday you can come here in Venice and hear about some really cool new stuff every weekend. I'm hoping we'll kind of accomplish that. And anyway, so excited to be here today to talk about AI for protein engineering and precision oncology with my dear friend Vivek. And maybe we can get started if you want to introduce yourself and your work a little bit.
Yeah, absolutely. So I'm currently a research data scientist at the Cedars-Sinai Hospital in LA. And my work kind of centers around helping to build software primarily in two major areas in cancer research. So it's helping to build out platforms that can kind of introduce optimized patient screening, so matching patients to clinical trials. And then the other major area is in multi-omics kind of analyses pipelines. So I'm helping to build out the software for that and primarily focused right now on RNA transcriptomics analysis. And this is in the setting of breast cancer liver metastases. And we're focused on liver mets in particular because they tend to be more immune cold or more resistant to a lot of different drug therapies. So we're trying to analyze our patients' RNA from their tumor samples to better examine the tumor microenvironment to see if we can isolate any specific immune cells or specific mechanistic pathways that we can then target for future therapeutic intervention. So yeah, in getting more involved with that work, I kind of connected with Jerry and Stanley and everyone here, and yeah, it's been incredible. I've been learning a lot more on the computational side being here as well, because I think the next phase of advancement in cancer research and just in bioinformatics in general is going to take you know, this kind of quantum leap forward in our ability to analyze these increasingly enormous data sets. Yeah, if you take a look at even one patient's complete set of genetic information, their genome or their transcriptome, it's an astonishingly huge amount of data. So I think these more advanced computational techniques and machine learning are going to be absolutely key to helping to solve cancer and a lot of advanced diseases. So yeah.
Oh, heck yeah! And we're gonna go through this presentation that I hope is gonna unpack some of these big words that we use in discussing this field, because it does get a little complicated. Yeah. But it's you know, proteins, transcriptomics, genomics. It's how life happens. It's how our lives happen within our bodies. So I think it's something that's really important for, you know, us to learn about, even if we're not sort of scientists in the core part of our life. Especially because some of the technologies we'll be alluding to in this presentation are really going to start impacting people day to day. And there's going to be a whole new layer of health decision making that relates to the data that you have about your condition and how people are going to use your data to create precision targeted therapeutics. Yeah. So I'm just really excited, and we're going to dig in. And then just quickly on me, my name's Stanley Bishop. I also go by Science Stanley. I'm a mathematician who mostly works on the computer science side of bioinformatics. So I'm really passionate about technologies that help people like Vivek do their job better. And then one little note of context too. So the Cedars Oncology group that Vivek works with, I don't want to embarrass you, but these guys are the Harlem Globetrotters of oncology. So these are the people you would want working with one of your family members if something ever happened to them. And so just really exciting to be here with Vivek sharing some of the ways that AI is really changing this field. But yeah, we want to get into, first of all, what is a protein? Because we hear about protein and we often hear about it in the Ike the Giant context. Since we're talking about proteins, I decided to invite one of the world's biggest bodybuilders, because who would know better about protein synthesis than Ike? But we're not just talking about the proteins that you eat. we're talking about the proteins that are actually nanoscale robots that run our entire biology. Is that too weird?
No, I think that's a fantastic way to describe it. Yeah, yeah, absolutely. Yeah, and then we'll get dive deeper into the different levels or the different functions that proteins can take on. Yeah, I guess if we can go to the next. Yeah. So overall, how do you get to a protein, right? So you follow the central dogma of molecular biology is you go from DNA, which gets transcribed into RNA and gets translated into protein. And overall, I think a great way you can think about this is it's how information flows from your genetic code to the molecules in your bodies that are proteins that actually do things. So these are the components of your cells that actually make things happen. And I thought this point was kind of interesting. It's something that I've been thinking a lot about. And if proteins are constructed from a language, which is going from nucleic acids to amino acids, then proteins contain information and functionality. And another kind of interesting spin-off of this is at the RNA stage, at the middle. There's something called a ribozyme. And in this way, you can think of RNA as not only holding your genetic information or the blueprint that makes you who you are, but they also can enact a lot of these actions and take on some enzymatic functions too, which is really interesting. So yeah, not only do they code for their own information, but they can contort and fold and carry out reactions themselves. But proteins are, the specialized effector molecules in your cells that actually do things. And all enzymes are proteins. Yeah.
And just to, because for me, as someone trained as a mathematician, this bio stuff is something I've had to learn in the field. So I do my best to think about it the right way, but it's sometimes challenging. And I know there's a lot of tech people in the audience, so I just want to introduce this metaphor of this bioinformatics cycle where information stored in DNA becomes RNA and then folds into a protein. You can almost think of it as a compiler cycle in a computer. And in a computer, we have our programs stored as ones and zeros. Not dissimilar with DNA, right? DNA has 4 letters. So it's not a binary sequence language, but it's a quaternary sequence language. Yeah. And it isn't a one-to-one metaphor, but it actually is quite close. That, we can almost think about the DNA as the hard drive. The system goes and gets the information in the hard drive, puts it in the RAM so that it's ready to run.
Yeah.
And that would kind of be like the RNA. And then when it's actually doing the thing, that's when it's the protein. I know that's butchering the science a little bit to look at it that way, but...
No, I think it's a very accurate analogy. And the reason I've also asked this question when I started learning about this is why is our information even stored in DNA form if it has to kind of come all the way down even through RNA and then to your effector molecules? And there are a lot of protective, your DNA or your genetic information is, you can think of it as extremely precious, right? So when it's in its DNA state, there are a lot of mechanisms at play that kind of protect your genetic code. Say like if you get exposed to radiation, we get exposed to cosmic radiation, UV, etc. A lot of mutagens that we get exposed to that kind of introduce these little mutations, also referred to as SNPs or single nucleotide polymorphisms in some cases. And you have very accurate, very efficient DNA enzymes that go in and repair all of these mutations all the time silently without you even realizing it. And all of us actually develop cancer cells in our bodies all the time, but your kind of protein machinery in your body is so good in general at being able to find these rogue cells and killing them that you would otherwise never know it. Yeah, it's just sometimes these rogue cells happen to get past our protein machinery that ensures our genetic code is still what it's supposed to be, and that's when you have development of cancer. So yeah.
I'm going to try not to belabor these bioinformatics versus computer informatics like metaphor too heavily, but it's almost like when you have a hard drive, you back up your data, or you have like a RAID array. And so very similar, it's kind of like DNA is oriented to be that long-term store informatically. RNA is all ready for runtime. And then the protein is when the program's actually running. And then right here, we got a kind of cool example of one of the important proteins. This is a hemoglobin, which exists in our blood, right, and helps our blood move nutrients and oxygen.
Absolutely, absolutely, yeah.
And so you can kind of see that the shape that this protein's folded in allows these little disky type things to fit into it. And so the shape of the protein, and again, I'm figuratively thinking of the protein as a little robot that has been designed to move these things around our bodies. It happens. It just, the RNA is laid out, and then it folds up in on itself. And so, again, these are self-building little robots that run our whole lives. I love it.
Absolutely.
Hemoglobin is a metalloprotein, so what it's holding on to, those little discs, are iron ions. And so yeah, you can have metalloproteins interact with calcium, and interact with other things. So just like we have robots that serve a purpose, same applies in this nano world.
100%. And then we'll be getting into some of the cool details and complexities of these systems. But it really is something I hope you guys will find staggering, the diversity of functions that proteins can perform. And beyond understanding how proteins work in our body as a revolutionary thing medically, we are likely to see many different industries disrupted by custom-built proteins that do tasks better than any technology we've built before. This is a field of synthetic biology, and the core tool that we'll be reviewing at the end of the presentation is going to be a big deal in synthetic biology. Anyway, I thought that was a good first slide. What do you guys think?
Oh yeah, I think so too.
So here we go, though, just getting into what we were talking about, the idea that this sort of bioinformatic runtime can produce many different crazy protein robots, right, Vivek?
Oh yeah, absolutely. And yeah, and the kind of realm of proteins, it permeates every conceivable organ system or area of your body. And I think that one of the key takeaways from this presentation, if there's only like one or two things you can take away, let one of them be that folding determines function. The protein's folding pattern is absolutely critical to determining how it's going to act and how it's going to function.
So
yeah.
As an example for the hemoglobin, it has little pockets that the things that it carries around fit into. And so the 3D geometric structure of the protein 100% determines how it functions and operates. And because I mentioned synthetic biology, I'm definitely going to point to this little spider silk example. So spider silk, which is one of the most tensily strong substances discovered, is a protein. And so in the spider genome, there's a little sequence that tells the spider silk protein how to shape itself. And here's something that we're just learning to do that's pretty incredible. We can actually take that sequence for creating spider silk, and we can put it in yeast. And the same way that we use a yeast bioreactor to create alcohol, which we call beer, we can use it to create spider silk. And so in the future, Kevlar vests might be grown in bioreactors using yeast, and other construction materials will actually be created using synthetic biology. So it's beyond an enormous area of impact for our health, it's an area that will probably start to affect many different industries. So pretty fun to talk about. Anything more on this slide for you, my friend?
Yeah, no, to that point, I think, yeah, it's important to emphasize that because protein is born out of this collective language of life that all organisms on this planet share, one of the most intriguing and impactful things to note is that you can take enzymes or molecules that you can find even in microbes that evolved long before we did, and we can adapt them in ways that would be helpful to us, either introducing them into our bodies or in industry kind of adopting some of these enzymes like you would find in archaea, which are very heat resistant. We took their heat-resistant enzymes and created something called PCR, polymerase chain reaction. And that's how actually we can take a very small sample of some organism's DNA and amplify it, create so many copies of it that we can then use that to run useful experiments, and one of them being in forensics. So that's how you can go from finding a tiny sample of someone's DNA at some crime scene, for example, and use PCR to kind of amplify their DNA so that you can identify who they are. So that enzyme actually came from archaea. So yeah.
When you say amplify, do you mean like visually or energetically? Thank you.
No, it just means to literally create more copies of that DNA that you found.
Oh, okay.
And yeah, to run a lot of experiments, to do research and to identify, just basically to be able to run very high throughput assays and experiments, you just need enough of that DNA material present in order to make conclusions.
It literally kind of just creates many, many copies of the sequence that you give it, and then you use the fact that you then have all those copies to study what is in the copies. Yeah. So you could almost think of it as like, you know, your trash folder in your computer has so many different files, it's hard to find the one you need. Yeah. But if you duplicate that file 10,000 times, you're probably going to find it pretty easily. So yeah. bad metaphor again. But yeah, and it's almost incredible because it's like there's this microscopic world down there that is such a huge part of everything that happens in our lives. But until recently, like we could barely get a glimpse of it, and we had almost no tools for actually interacting with it. And we're at this moment that's the start of us having those tools to design proteins in a precise way with specific functions and really kind of understand in a deep way what's going on here. I want to throw out one more protein
one quick last question on
please.
The PCR.
Yeah.
Is that the same PCR as from the COVID test?
Yes, correct. Yep. Yeah,
and then that's exactly what happens, right? Is you get a little fragment of the DNA and then you multiply it and...
Yeah, absolutely. No, that's a great point. And I think that's another example showing that any tools that you can find in one domain or realm of biology can be adapted to so many other areas. And yeah, it's just each one of these techniques you can think of as a technique in our overall tool belt, and it's up to us to get as creative as we can to apply it to as many scenarios as possible. So yeah,
it's kind of fun to think of it as a tool-based thing too, because I was an electrician's assistant growing up. I worked on a lot of contracting sites, and it's kind of like, you know, if you show up to do an electrical job and you just got a screwdriver, you're not getting anywhere. But it's kind of like when you start adding tools, it gets easier and easier, and then you get to this point where you can actually work on a complex system positively. And again, I think we're just at that point in this field, so it's very exciting. Wanted to share one other example, because this is a protein we'll actually be looking at just in a moment or two. But there's a bioluminescent protein that's very similar to proteins that exist in many other organisms. And so you can actually change the sequence so that instead of building itself with normal tissues, the animal builds itself with bioluminescent tissues. And then you can study the shape of them based on the emissions.
Yeah.
So it's just an incredible moment where it feels very creative the way people are using all of these new things we're finding about engineering protein function. And then again, here's just to really hit it. Proteins are self-building robots, you guys.
Yeah, I know. And yeah, I know it looks like there's a very busy slide, but basically this is just one example of how you can kind of exploit the self-building characteristic of proteins to create some really interesting structures that you can use for various functions. And yeah, in this case, the example is creating a nanocage via the allosteric effect. And what allosteric means is you can have a protein and it has its main binding site or its main binding pocket, but off to the side, it'll have another secret binding site called allosteric site, and another ligand or another molecule that can fit into that site. Once it does bind to that allosteric site, it can completely change and contort the conformation or the folding of the protein so that it can then kind of quote unquote activate it and allow it to bind to another ligand or another receptor molecule or something like that.
So forgive me, but I just want to share that ligand just means molecule. Oh. And again, the idea is that sort of these proteins interact with each other and with molecules, and then that dance of interactions is what we call life. Mm-hmm. But then again, the specifics of a protein's function comes down very much, like Vivek's saying, to its geometry. So it's kind of like in this situation, you have a protein that's set up a certain way. It has like a little key slot though. And if the right molecule fits in, the whole protein shifts and then has a different function.
Yeah, exactly. And it's that that phenomenon, that allosteric effect, that is one of the integral components that allows you to make this kind of a structure. And I think one good analogy that I have for this is I like to think of this characteristic of self-building robots and proteins as drawing a lot of similarities to polymers in industry. So like if you're making plastics, for example, you take a lot of the same repeating chains of a molecule, and if you attach them all together, you get a polymer, and one of which is plastic. And so you can imagine if all the various shapes and functions we've been able to achieve with plastics or polymers in industry that are applied to that are now everywhere in our daily lives. Imagine if you can apply that same set of diverse end results or functionality to the biological world. So you can quickly start to see that there's almost limitless capability for what types of nanostructures that we'd be able to build from proteins as well. So,
100%. And it's actually one of the really interesting things. There's a way mathematically to approach thinking about the space of all possible proteins that could exist. And it is shockingly large. Like it takes like a 20-minute math lesson to understand how large it is. Like it's one of those things where the number of possible proteins surpasses the stars in the sky kind of vibration. Ed, question?
Yeah, I was wondering about the protein interaction. Do they typically work in concert with other proteins to accomplish their goals, or are they more solo purposed?
Oh no, absolutely. So it's something that's not included in this presentation, but if you look, you can Google this, but you can see kind of like a mosaic or a chart of all human biochemistry. And it's this ridiculously complex set of pathways that all interact with each other. And there's still so much left to be discovered also. So yes, absolutely, they all interact with each other. And that's actually one reason why we have to be so careful with CRISPR and gene editing, is because if we want to introduce a specific kind of functionality or change into our biology to treat one particular disease, we have to be certain that the change that we're introducing is not in some deleterious or negative fashion affecting all of these other critical pathways in our biochemistry. So yeah.
There's really an enormous amount of nonlinearity in how all these things interact within the system too. And then CRISPR, which you just mentioned, people haven't heard of that. That's this gene editing technology where essentially you can kind of take a certain sequence of an organism's genome and switch it out for a different piece. So as I was saying, like we can take spider silk from a spider and then get yeast to grow us that material by just kind of swapping out. So anyway, such cool stuff. Should I maybe go to the, this is such a great slide. Oh, Ed, go ahead, sorry. Yeah, of course.
So to be more specific, are the proteins often working in concert with each other to serve a specific task?
They very much are. Sometimes in the sense that actually a structure could fold from multiple proteins, and so we'll actually be looking at a couple multiplex proteins where a certain shape is created by multiple proteins growing together. And there are quite literally proteins that are electrochemical motors. Like literally, you look at the thing and it looks like an electrical motor, and it basically has one protein, another protein around it, and then it's set up so that one protein can spin due to an electrochemical force. So there's quite a bit of complexity, and then there's quite a bit of complexity between different proteins fitting together and causing different things to happen. One of the things that I'm very excited about with these technologies is using some of the stuff we'll be talking about to find medicines to treat rare diseases. I maybe we'll keep that for a later slide to really dig into, but we will share some more Ed about sort of the sort of complexity and nature of protein-protein interactions. Next slide. What is an oncoprotein?
Yeah.
And Vivek, I might let you start this one. I realize I maybe have to hit the bathroom real quick if I'm going to do
Go for it
on this. So just tell them about oncoproteins. I'll be right back.
Okay, sure thing. Yeah. So for oncoproteins, Basically, what they are, I guess more broadly, we can think of them as all the different types of protein that can be involved in cancer, or in the proliferation of cancer, in oncogenesis. In the realm of oncology, it's one of the primary reasons we haven't found a cure per se for cancer is because of the staggering complexity, not just in pan cancer, but there's an incredibly large number of differences, even just going from one type of cancer to the next. And yeah, if I can see, yeah, so here for example, you can kind of see a network of proteins that are involved in certain types of cancers and their relation to each other. And this is just a small picture of the overall picture. And yeah, this also gets staggeringly complex. At least for examining this case, I guess we can focus in on a few proteins, and we'll be doing that as well as part of our code and demo later. But some proteins are directly involved in oncogenesis, and those are oncoproteins that we're specifically talking about. And then some classes, some major classes that also happen to be major therapeutic targets in oncology involve immune checkpoints, different effector cells that control cancer metabolism, and genomic regulation also. And I think Stanley will touch on this also. It's an area that both of us have been very fascinated with, and that's with introducing viruses into the world of cancer. So yeah, in general, you can think of proteins in oncology is that there's a very intricate related network of proteins that play off of each other and can modulate each other up or down, or make other molecules that are present in this pathway go quiescent as well. And that can create some issues for our bodies that make it more difficult for us to fight the cancers that we can develop. And in particular, one area that I think a lot of researchers, and I think rightfully so, are going after are these universal mechanisms that are common to all different types of cancer. I think this is going to be one protein that's also going to be involved in our demo later, but it'll involve TP53, which helps control a lot of our cell apoptosis and is one of the central proteins involved in, that's actually mutated. The gene that codes for that protein is mutated in virtually all types of cancers that we know of. So yeah, absolutely.
So I know in a lot of cancer tests that oncologists give to determine efficacy and whether you're getting treatments or working stuff, they're looking for the protein markers. So these are some of the proteins that they might be looking for.
Yes. Yeah, absolutely. So and you can test for these proteins in different ways. You can do a tumor biopsy in different forms. So you can get a piece of the tumor tissue. You can get a DNA sample. Oftentimes for patients that are at very high risk of developing cancer, either they have that genetic germline history for it, or they've already developed cancer and they've treated it, and now you go through normal surveillance, and so you'll get regular blood tests every 6 months or so. And so there are particular types of molecules or proteins that will set as a molecular biomarker for cancer in general, specifically for their type that they'd be at risk for. And you can get regular blood tests, let's say at 6 month intervals, and you can check the levels of that molecule in your blood. And if it happens to deviate from its normal level, like if it gets a little bit elevated, then that can be a very reliable indication that you have some kind of oncogenetic process occurring in your body. And at that time, it would then be prudent to kind of test for different types of cancers that person would potentially be at risk for. So yeah, but yeah.
Sorry, wireless mic is dead, so you'll
Just make us feel good.
and we won't have chords, we won't hear beautiful voices.
Yeah, park it near Ed, man. He's had some good questions, and sorry for stepping out, but pretty incredible. As you can see here, the complexity of the network of interacting proteins and molecules, and so it's a pretty special moment. We're starting to just have the tools to understand how all this stuff connects.
Yeah, absolutely. And and this also, I think, helps drive home the point that any change you make in one area can have a profound effect in a lot of different other areas. So I think part of this next phase of research that's occurring, and that we'll definitely need the answers to, is what other pathways exist, and how do the proteins or genes that we currently know of, how will they affect those other pathways? So yeah, I think that'll definitely be one of the next major areas that we're going to have to understand a lot better. So yeah.
Incredibly interesting. And now multi-omics.
Yeah, so multi-omics is, I would say, currently the best way that we have of studying cancer in general. And this heavily relies upon studying proteins as well. So genomics involves analysis of DNA, transcriptomics for RNA, proteomics, proteins, and metabolomics involves all the pathways by which all these molecules interact with each other. So by studying and kind of correlating all the findings that we produce from each one of these subfields in our analysis, we can kind of bring all of that together with a lot of computational methods to then elucidate exactly what's going on in a specific part of cancer disease called the tumor microenvironment. And this is basically what you find when you zoom all the way in and see all the cells that are adjacent to your cancer cells in your body. And yeah, I think that this is a pretty good mosaic that you can see on the right side. Where you have blood vessels that come through and give nutrients to that tumor, and you have some malignant or tumor cells present, as well as some of your immune cells, some of your healthy tissue, a lot of signaling molecules that are there as well. So as you can see, this kind of gives a very cursory window into the incredible complexity that is involved with any type of cancer. So,
it's kind of funny word too, multi-omics, right? And it's because we got lots of omics. We got genomics, proteomics, metabolomics, and yeah, multi-omics is this idea that if we layer all of that data on top of itself and we sort of study the patterns, that like we can start to work out this really high detailed granular resolution of like what interactions are happening between the tumor and the surrounding tissue very specifically. And then, here's the question. Can we maybe find the key specific interface between the tumor and their surrounding cells that is sustaining the tumor? And can we cut it? Can we start to target more precisely the metabolic and biochemical functions that are keeping a tumor alive and sort of, turn that off without affecting the entire patient's metabolism? Oh man, this is such a great picture too. It really kind of captures how much stuff is flying around. You have a question, Ed, or?
I do. So traditional chemotherapy, is that then attacking the microbiome for the tumor itself, and that's why it kills so much of you? And
yeah. So chemo, you can think of more as indiscriminate carpet bombing of the cells in your body, and the basically the overall goal, which is a little bit brutal actually, is with chemo, you want to administer enough of it to a patient such that it'll wipe out all their cancer cells before it gets to a point when it wipes out all of your other healthy cells. So that's why traditionally chemotherapy produces such toxic effects and it's so difficult to go through. And that's why approaches like with multiomics is so critical, those approaches are so important because you want to selectively target only the tumor cells and leave all your healthy cells alone. So, yeah.
right, so there are dozens of chemotherapy drugs, right? Are they each one's targeted towards a specific microbiome for a specific type of tumor?
Yes, yeah. So the different chemotherapy drugs do target different mechanisms. Some of them, you can target specific proteins that are present on the surface of your cells, like certain receptors. You can target. I know a major target is, so say you fundamentally need to ask, what is cancer? It's a cell that has gone rogue, and now it's just dividing uncontrollably. So then you can focus in on the mechanisms that are central to a cell that is dividing. So what is absolutely required for your cell to divide? One component is a protein called microtubules. And microtubules are, they're involved, so when you have a cell that's getting ready to divide, its nucleus, or all of your genetic information, is going to need to replicate. And once it does this, it kind of moves to different poles, opposite poles of that cell. And as it's doing that, it forms a connection between the two sites, or your two new nuclei for your future two new cells. And the connections are formed by microtubules. So if you, so then it wouldn't be hard to imagine, if you want to shut down that cell's ability to divide, all you need to do is disrupt that microtubule network. And that's one type of chemo drug targets microtubules. But as you can imagine, your healthy cells also need to divide, so they also have that protein present. And so that type of chemotherapy will also target your healthy cells. So now you can imagine why it's so important to come up with other targets that are specifically only present on your tumor cells and not on your healthy ones. So yeah.
And actually, at the end of the presentation, we'll be inviting you guys to fold some of your own proteins. And we've actually selected a very key immune protein that's kind of used by our immune system to target a particular virus or bacteria. And then this is actually being used in a type of treatment called an antibody drug conjugate, where you literally, Ed, you're going to love this. You take a chemo molecule and you attach it to an immune cell so that the immune cell delivers the chemo molecule directly to the target site. And so actually, like, this is a field of therapeutics that's kind of taking steps towards being something you could call side effectless chemo.
Yeah, precisely. So through this mechanism, or through this paradigm, you can take a toxic payload, your toxic chemotherapy molecule, attach it via a linker molecule to an immune molecule. Like in this case, it's going to be an antibody, and antibodies, are already present in our normal bodies, but our immune system normally produces these to fight foreign pathogens that enter our body. But in this case, we're kind of just hijacking or borrowing our own machinery that we have present in our bodies anyway, in antibodies, and using that to target cancer cells that are presenting a specific type of antigen that those antibodies bind to. And in this way, you kind of ignore all of your healthy cells that are not presenting those specific antigens. And that in this way, you can deliver a toxic chemotherapy molecule specifically only to cancer cells, just by using a part of an immune system that we already have. So, yeah.
And if you know anyone who's gone through chemotherapy, it's hard to overstate what an exciting prospect that is. And that's just one prospect for these new treatments that take a precision approach to attacking the tumor without targeting the whole body. And then again, just to return to that metaphor of, like, earlier we were on the job site with just a screwdriver, and now we're sort of like getting closer to having tools where we can really make a difference. Lance, did you have a?
Yeah, I had one question. Is there any way of identifying the blood vessels that go to the tumor? And are they feeding the tumor and everything else? Because couldn't you just cut off the blood to the tumor?
Yeah, that's a great question. And there are, so especially if you have a very vascular, huge tumor that has, tumors, you can actually think of them as having their own pseudo brain, because they're very good at figuring out exactly what they need and kind of hijacking our own physiology for their own benefit. And one of them is recruitment of blood vessels, right? Because they want to give themselves as many nutrients as possible, even to the detriment of the overall organism that they're growing in. Like, they don't care. So in those cases where you have a highly vascular tumor, or a tumor that has a lot of blood vessels going to it, it then becomes advantageous to administer a type of drug that cuts down angiogenesis, or production of new blood vessels. So yeah, if you were to administer a drug in a vascularized tumor scenario, then you could presumably cut down that tumor's growth. So yeah.
And it's kind of interesting, the intelligence of a tumor, because like it doesn't technically have a brain, but like kind of what happens is, as Vivek mentioned, our bodies create thousands of mutations that could become a tumor. I don't know if I should say every day, but probably every day, right?
Yeah.
But it's sort of, in order for the body's systems that are that exist to suppress tumors and correct tumors, so many things have to go wrong for it to sneak through those systems, that it's sort of like the ones that do are the smartest tumors, or the ones that have figured out the right configuration of co-opting the body's systems. But anyway, I have to say, I'm stealing this image for future presentations, man. This is so good.
Oh, no, for sure.
And that was a lot, so definitely let me know any questions, but I think maybe ready for the next...
Yeah, let's do it.
And here we go. Here's where it's really interesting. We're starting to wonder about because we're talking about proteins as the sort of how things actually happen in biology. So is it going to be possible for us to understand the proteins that a tumor uses specifically enough to target it? Is that the question?
Yeah, absolutely. So basically in this way, the next phase is then to identify or classify the types of tumors that are present based on the proteins that they have. And one canonical example here is with breast cancer. So I would say classically there are 3 major types of receptors that we would test for if a patient develops breast cancer. So is their tumor hormone positive or negative? And hormone meaning ER or PR, which is for estrogen and progesterone. And then the last one is going to be HER2. So basically why we would test for these specific types of proteins is if we can test and see that a breast cancer patient's tumor is positive for any one of these types of receptors, then we then know that their tumor has these targets present. And we can then use these receptors as targets for therapeutic intervention. And there are specific drugs that have been developed to go in and end up killing their tumor cells if they have like estrogen receptors still present on their cells, for example. And as you can then imagine, it becomes much more difficult to treat and deal with if they're negative for all of these receptors, if they don't have any of them present on their tumor cells. And this is what's referred to as triple negative. So triple negative breast cancer, TNBC, as you've probably heard of before. And then, so in these scenarios where you're missing a lot of these common targets for therapies, it then forces you to become a lot more creative and go hunting for other types of mechanisms or receptors that you can potentially target. Because their specific tumor will not respond to all of these other therapies that we've created to target these other receptors. Yeah, and another interesting kind of a mechanistic approach to therapy is if their tumor does not have some of these targets present, can you use something like a virus or some other vector, some other type of gene editing to then go into their tumor cells and augment their genetic expression to start expressing that specific receptor that they lost, so that you can reintroduce this target into their cells and then start attacking it. So yeah, that's another mechanism by which you can potentially treat tumors. But yeah.
And again, this I feel so lucky to be hearing you share this wisdom, and thank you so much for being here, Vivek.
No.
And yeah, just to underline, there's this feeling of like, it's like we were trying to fix computers, but we didn't know about computers or software or hard drives or anything. And it's like now, we can open up the machine, look at the pieces, and we're starting to really learn how they come together and what we can do with them. And yeah, why do we care? And I love this slide. I'm going to share at the end of this talk one of my favorite YouTube videos, and it's by a YouTube channel called Kurzgesagt. Any Kurzgesagt fans in the house? Oh, hell yeah, guys. Everyone, do me a favor, check out this YouTube channel when we share the video. This is just some of the best science education in the whole world right now. It's cartoon driven, but the level of communication and the technical details are like very high and rigorous. And they have this one video that talks about a medicine for precision, a metaphor for precision oncology, where you imagine that a tumor is like a city that's gone rogue. Like maybe the criminals have taken over the city, or the mafia has taken over the local city government. And the reason why we imagine the city as a metaphor for the tumor, because economics is one of the only places where we see complex interlaced systems quite like this. So you imagine like a city, how many imports and exports does a city have? You know, the total diversity of the flow in and out. And it's like the tumor micro environment, the sort of the net of all the metabolic interaction, genomic interactions between the tumor and the surrounding cells. It's just actually very close to this little image of a rogue tumor city that I generated in DALL-E. And then I also love this image you added, Vivek, which is this personified tumor microenvironment. Can you tell us a little bit about these characters here?
Oh yeah, absolutely. So yeah, the cell that's kind of trying to pierce the main tumor cell in the middle is that NKC, or the natural killer cell that you see at the bottom left, also up top there, yeah. And then you have different types, major types of immune cells that are involved, not only so in fighting cancers, but anytime you get an infection, for example, these same types of cells are present here. So you can kind of see your body looks at tumor cells much in the same way that it would look at other pathogens or foreign microbes. So yeah, as you can see, there's some of your B cells which produce some of your antibodies. Those get shot at the tumor. You have your major T cells. So both major parts of your immune system, which is humoral immunity, which involves production of antibodies, and cell-mediated, which involves T cells, for example. Yeah, you have different types of signaling that are involved: interferon gamma. You have CD4 helper cells, and all different types of immune cells that are involved in fighting pathogens, but also, of course, here fighting tumors as well. So.
I really like this co-opted regulatory T cell that's telling the natural killer cell to leave the tumor alone.
Yeah,
it's really. Yeah. I mean, I love cartoons, guys. I don't know.
Yeah.
But yeah, again, and it's just such an interesting metaphor. And then we think about like kind of what economic warfare looks like, right? Like we embargo countries, like we don't let them import oil because that's a key object, or we don't let them import chips because that's a key object. So, there's just a layer of interpretation where we might say we're kind of building a metabolic economic model for the tumor cell, where we can kind of strategically target and limit the things it needs to cause trouble. And that's what this slide is about exactly.
Yeah, absolutely. So now specifically getting into the realm of precision medicine, which also kind of crosses over with immunotherapy. And one prominent example that a lot of you have probably heard of is CAR T cell therapy. And basically this paradigm kind of involves going into a patient that has developed cancer, doing something called lymphodepletion, or kind of taking all of their T cells from their body, bringing it back to the lab, and genetically engineering the T cells to act in a certain way, mass producing them, and then introducing them back into the patient. So in this way, you kind of combine elements of your immune system and genetic engineering to go in and that would specifically tune their immune cells to then target their own cancer. So yeah, this is the part of the approach that imbues precision. This is precision or targeted medicine. And in this way, I think the next large phase of this type of approach, a lot of work actually at which is being done at Stanford. We've had some great investigators come over to Cedars and give talks on this as well. And that is kind of integrating real world, real-time data collection, like say with an Apple Watch or some other type of machinery, to then introduce real-time monitoring through telemetry of different types of molecules or processes that are normally occurring in your body. And so we can kind of switch to the next phase where we're not just kind of reacting once you develop disease, but we're trying to get ahead of the disease and prevent it from getting to a more advanced state. So we're trying to catch development of the disease in its very early stages. Like if we say, like if you're constantly monitoring your blood, say like using one of the infrared sensors, like on an Apple Watch, for example, and you've specifically tuned that infrared analysis for a particular molecule or protein that is acting as a molecular biomarker for major type or common types of cancer, for example, then whenever you would see that level kind of fluctuate up a little bit in your bloodstream, a red flag or an alert would go off, much like it would currently, like for blood pressure or for your heart rate, and it would go straight to your physician, for example. They would get the alert and they would bring you in for a more detailed blood test or something like that. And in that way, you can hopefully catch cancers that are extremely difficult to treat, like pancreatic cancer, much earlier, and that should help our health tremendously. So,
just to underline what we just said here, because I feel like this might take some interpretation. Because like here's what we're talking about, y'all. 10 years from now, 15 years from now, your kid gets their first smartwatch, and you say, sometimes you might get a notification on your smartwatch that a cancer has been detected, and then you just have to go into Kaiser, get an mRNA shot that's kind of customized to target the immune system at the cancer that's developing, and then it's done. And then, maybe someday your kid says to you, hey, what used to happen before we had Apple Watches? And then we get to say, oh, well, there used to be this thing called cancer that people died from. And it was actually a very common cause of death. And, thankfully, we got to a place where we didn't have to deal with that anymore.
Yeah.
And again, like interacting with patients day to day, week to week, there's a feeling of like not wanting to share that vision because like we don't know when it's going to happen, and we want hope to be rooted on reality for patients. But this stuff is genuinely coming together at such a fast speed. And in every direction of kind of data and interpreting and working with data, we're seeing revolutionary technologies. So I personally don't think that's a pipe dream.
Oh, absolutely. No, I think, we're starting to see the underpinnings of success in some areas. And I think it's very promising kind of seeing the incredible progress that's made in other disease types, in other domains in human health. Even if it's not cancer, but now we can, if we're starting to see some of these positive signals in other types of diseases, it's not hard to then imagine that we should be able to make the same leap in oncology also. But, yeah.
Yeah. How would you introduce those detectable markers into the person?
Okay, so it's probably going to take a combination of first identifying the optimal targets or the optimal biomarkers that we should be testing for, right? And in order to do that, we're going to have to further analyze, do these types of multi-omics analyses of cancers. And then probably that in and of itself is quite complex and tricky, but another added layer of complexity is of those new targets, or maybe it involves some existing target we've already discovered. But of all of these lists of targets, which one can you reliably detect with current or developing technology that we have that can reasonably fit in our existing health devices? So it needs to be cost effective. It needs to be sufficiently sensitive enough to detect that specific molecule. And does the current state of technology, like for infrared sensors, for example, is it at a sufficient enough state? Has it been developed far enough such that we can use it to analyze or detect certain types of molecules? So, yeah, it's going to take a convergence of multi-omics analysis, of identifying good targets, and then having the the telemetric technology good enough such that you can actually detect or pick up on those targets. So, yeah.
So if you have those, would it be like a shot you'd get once a year? Is it a pill you could take?
Yeah, I think it depends on the mechanism that you target, and I guess the downstream effects of that that type of mechanism that you would introduce into the body. So yeah, I think it would highly depend either on the type of disease that you're treating, and also the mechanism of action like for that treatment. You know, ideal scenario, it would be kind of a one-off thing, or maybe you get once a year or something like that. Maybe that is the eventual solution. Not sure yet, but yeah, it'll profoundly impact the cadence or the type of approach that we take for monitoring and treatment. So how often do you need to get tested? How often, does that mean you need to get treated? Is it every 3 months, every 6 months? Is it something that becomes analogous to how we think we're going to continue to deal with the flu and COVID, maybe like once a year type vaccine or something like that? I think that remains to be seen. So, yeah.
And there's just such a sort of ecosystem of different tools at each of these like kind of technological points that we're discussing that are emerging, and it's very exciting. You know, the cost of sequencing a genome actually falls faster than the cost of compute. It's actually a double exponential, whereas the cost of compute via Moore's law is just a simple exponential. And then also, like, we've really seen some things like mRNA vaccines where, you're actually harnessing the body's genetic machinery to produce a compound that's involved in a disease or vaccination process. And then we've started to see some other really interesting things happen with tools like mRNA vaccines. I don't know if anyone has a friend with a peanut allergy, but there's a cure for peanut allergies that has been used in clinical trials using mRNA vaccines to reprogram the body immunologically. So anyway, I just think one of the reasons we were so excited to share this talk is there are going to be all these things that are really important for us to be aware of for ourselves, and then also to make sure that these technologies are used in an equitable and positive way. Because, I don't know if we can trust big companies to always do that for us.
Right.
But yeah, it's such an exciting moment. And then, here we're getting more specifically into oncoproteins, because the kind of final part of this talk and then the interactive part will be related to some of the new AI tools that are allowing us to understand proteins in much deeper ways than we have before. But I don't know, is there anything in this slide that we didn't cover?
Yeah, I think we covered most of this already. And maybe this is a more clear representation of one of the elements that we talked about, which is ADCs, or antibody drug conjugates. So if you can see on the picture on the right side, the top left, that is an antibody molecule, that little Y-shaped molecule. And it's to that molecule that you can attach something like a cytotoxic drug via a linker molecule. And in systemic circulation, so while it's outside of your cells, going through your bloodstream, it's not toxic, ideally. And then once, so the antibody should be specifically tuned to bind only with the antigenic components that are presented on tumor cells. Once it gets there, it kind of gets endocytosed, so goes into the interior of the cytosol of the tumor cell. Then it decouples that chemotherapy drug with or your cytotoxic payload would then decouple from the antibody, and then it becomes toxic. And it's at that point, it kind of kills the tumor cell from the inside out. So yeah, and this would be the primary molecule that would be responsible for kind of endowing the chemotherapy drug with its targeting ability, and it's that antibody molecule. So yeah.
And just for emphasis too, because this will get into actually something we'll be talking about. We're thinking maybe a couple weeks after some less nerdy presentations, we might show another cool thing that we'll talk about, and then that actually relates closely to this piece here, which is in order to understand how to target the DNA of the tumor, you have to understand it in the context of the host DNA. Oftentimes tumors will have some extra junk in there that could either be part of the reason why it became a tumor or just kind of a coincidental effect of some other process. And so it's almost like a needle in a haystack kind of thing, but you can kind of find these these targeting needles that are kind of specific proteins to the tumor, and then some of these treatments can be structured around that data. So anyway, that's all to say that's a type of data that Vivek and I are really interested in, that I think we'll be doing some data science and machine learning around in coming weeks and months. Man, such good images for these slides, bro. I mean, just knocked it out of the park. Thank you so much.
Oh, no, for sure. Thank you.
And then now we're getting into the part that will sort of be what we're doing together as an audience, if you guys are interested in it. We're going to be talking about some computational protein engineering with AI, and we'll also be talking a little bit about the history of the protein folding problem. And this is a problem that is near and dear to my heart. I grew up in Palo Alto, right across the street from Stanford. And particularly, when I was growing up, the Panday labs at Stanford were some of my biggest heroes. These are the people behind something called folding at home. Has anyone ever heard of folding at home? Oh, of course, Ed has heard of folding at home. Well, so folding at home was this really special program where if you had a computer you weren't using all the time, you could put that computer on the folding at home network, and then the Stanford molecular engineering department would use your compute to do some of their folding problems. So, maybe one of these proteins that's interesting for cancer research, you put your computer on the folding at home network, you get sent the protein, your computer starts working on figuring out how to convert from the sequence to the shape of the protein. I was really into this because when I was 10, I would run this on my PlayStation 3. And that was actually the first time I ever learned about scientific computing, distributed computing, and it's been my life since then. So I always just think the computational story of protein folding is so interesting and special. But then the part to really emphasize is how challenging it's been historically. It used to be you needed what's called a distributed computing system or a cluster to do this. And that really literally means like you might have needed to hook up 100, 1,000, 10,000 computers to work on the problem, because actually simulating the protein in all of its shape and then actually figuring out how everything interrelates just required an enormous amount of memory and compute. And so this was the case up until just about 2 or 3 years ago, when a team at DeepMind decided that they wanted to try to use AI to solve the protein folding problem, and they did it. They did it.
Yeah.
My whole life, right? No, seriously. And a lot of people say, though, that if it hadn't been DeepMind doing it, if it had been a traditional scientist, that person would have won the Nobel Prize. So it really gets into this interesting moment where all of a sudden AI has all these things to say in a field where it used to be very much just biologists saying things and discovering things. And anyway, beyond just the fact that they solved it, and I want to say something about what it means that they solved it, because there's actually something, it's the Olympics for protein folding nerds. It happens every year. It's called CAS-P. And basically teams from all over the world compete with different algorithms to try to get the highest percentage accuracy on some protein folding benchmarks. And so for my whole life, it's been one of my big excitements every year is I'm like, oh, the CAS-P results are out, and I'm like, oh man, the Harvard team got up to 82% accuracy. That's killer. They increased it a full 0.5% from last year. This is incredible, revolution. And then 2 years ago, DeepMind releases this technology called AlphaFold, 99.9% accuracy on every protein out of nowhere.
Yeah.
It's just, it's going to go down in history as one of the most remarkable scientific stories of all time. And so that's AlphaFold, and that's the main thing we're going to be talking about. We're actually going to be showing you guys, we have a Google Colab notebook we're going to share with you in just a second. You'll be able to log into there, put in a protein sequence, run it, and then you'll actually see the 3D structure. And then we also have a number of kind of interesting, fun proteins for you guys to try and learn about too. Did I see a hand? Sorry.
Do you guys know much about how they trained AlphaFold? Like, what was their training data? Or
We do know, I know quite a bit and have worked on some of these projects, and some of that is indicated down here in this architecture diagram. If it's okay, I'm not going to get into too many of the details, but suffice to say they used a large database of known sequences and then folded structures, and then they used deep learning technology. And then, like I said, if people are interested in digging into some of the more machine learning architecture details, like I would even be very happy to bring down a whiteboard when we're done and we're sort of folding proteins and talking. But the thing that I do want to emphasize before passing it back to Vivek for kind of his take on AlphaFold is just this graph here. So you can see AlphaFold announced December 2021, and then this is the exponential growth since then of papers published that use AlphaFold. Is it fair to say that this has kind of revolutionized the whole field of protein engineering, or?
Oh no, absolutely. I mean, I think very deservingly so. Often been referred to by a lot of people as the biggest breakthrough in biology since the Human Genome Project. I mean, it's that significant, and the importance of AlphaFold, just to kind of drive home its significance, is this platform allows you to predict with a very high degree of accuracy the three-dimensional structure of a protein just from its amino acid sequence. And that is absolutely incredible. It's critical how you can just take a sequence, or just a sequence of letters or residues, amino acid residues in a language, and be able to infer how this polypeptide or this protein is going to fold itself and then endow its specific function. So yeah.
To bring home too, like given this metaphor of like bioinformatics and computer informatics, like if some of you guys are coders, imagine if you had to code without being able to compile your code and test it, see if you're going up the right. Imagine like you cannot run your code at all until you are done, and then you have to say, I'm done, I can't change anything more, and then at that point you get to test it. Yeah. Like this just opens up the ability for people to try a protein, see what it does, change it a little bit. And then just one example of like where I've been using AlphaFold in an interesting way. Has anyone ever heard of the avian flu? So this is a flu that we're very concerned about because while it usually doesn't infect humans, when it does, the mortality rate is over 80%. And so we're very concerned with predicting and hopefully preventing a zoonosis of this virus. Zoonosis is the transfer to humans from an animal reservoir. And it actually all comes down, again, to the shape of a protein. And it's a question of like, is this protein in a shape where it can attach to a human lung? And again, it's sort of like we can start with just the sequence. We can predict how the sequence might change, and then we can actually see what consequences that has. And then in that case, like we sort of know what to look for. And we can maybe start to do things like developing vaccines before a virus evolves, and so that we're ready for it. But anyway, so this is the kind of intro to AlphaFold. And then we wanted to get into what's next before we actually turn you guys loose on AlphaFold. And we're actually hoping, too, that we might be able to get a second bite at the apple, tell you guys some more nerdy stuff in a few weeks or a couple months. But yeah, future of AI-driven protein engineering is poised to revolutionize the field. And then in particular, there's some emerging technologies that enable the computation of not just structures, but interactions between proteins and different molecules. And again, Bosch, I think spiritually, your incredible talk on diffusion last week inspired us to pull this talk together. But then also, we're using the same machine learning algorithm here. The diff in diffdoc stands for diffusion. Some of you guys might be familiar with Stable Diffusion or Dolly or Midjourney, these like incredible technologies where you give them a string of letters and then they produce an incredible piece of art. Well, what if we do the same thing, not asking it for art, but asking it to predict how a given protein and a molecule interact with each other? And it turns out we can. And so there's this tool diffdoc. And similarly to AlphaFold, the docking computations where you actually understand how a protein and a molecule interact, they used to take big computers. Like we're talking university basements filled with 100 Mac Pros or 10,000 Mac Pros kind of scale. And now you can do it on a single GPU. You can do it with a single Colab notebook. You can actually compute some of these interactions. So yeah, I'm hoping we'll be able to discuss that more in the future, share that, explore that more in the future. And then the really exciting thing about this is it's kind of like, many illnesses haven't really gotten research into drugs that could help them. Unfortunately, a lot of these companies only allocate resources when there's a very substantial 1,000x, 10,000x profit potential. So there's all this like low-hanging fruit that hasn't been all these illnesses that maybe they afflict many people, but not enough for it to be very profitable. But the search hasn't even been done. And so the potential exists for, using machine learning, using all of the GPUs that are out there. If we could get 1% of the GPUs that all these big companies are buying and hoarding to be working on rare illnesses, like we might not have rare illnesses anymore. So anyway, it's just so exciting how this stuff's coming together. And I think this is our last slide. Is there anything about what, and forgive me, I'm kind of rushing through the last slide because I really want to get you guys the Colab notebook and get us folding. But any kind of final thoughts on this one, or?
No, I think you hit all the major points. I think hopefully everyone here will see just how much of a staggering tool or platform and advancement that AlphaFold and all of these protein modeling platforms or technologies are. How important they're going to be going forward, and I think that the key will be, I guess, to see how you can envision this helping not only others but also yourself, because I think this has an application, to better everyone's lives. And so just have fun with it, explore, and if you have any questions, we're here. And I think the tools and the demos that we have are going to be really fun for you guys to work with. So yeah.
Yay! And woo! And I think maybe hopefully Jerry can get it shared to the Telegram chat, but happy to share this in some other way too. But here we have a Google Colab notebook prepared for you guys. Hack Sunday AlphaFold. Jerry, how should we?
It's on both. It's on the Telegram, and I also put a little link on the website just in case.
Okay, cool. So if you guys go to hacksunday.com, you should be able to find this notebook. And I'm just going to walk through it like real quick and simple, and then we can all dive in together. But to start out, just kind of an intro to this talk, what we're doing here. This is a community-driven place where we're trying to learn together and kind of share what we all know from different fields. Here's a fun picture I did of a GPU rig folding some proteins. Go over a little bit of like what is in this notebook. We're talking a little bit about proteins and their function. We're talking about AlphaFold. Then we got some stuff about AlphaFold, what it is, how it works, how it's used. And then if you go down to here, you'll see a little section called interesting proteins and their biological functions. And I've selected 10 proteins. So for example, we start out with the hemoglobin, which was in the first slide of the presentation. So this is the protein that's responsible for moving stuff around in your blood. And then right down here is the sequence for hemoglobin. So if you want to fold hemoglobin, come down to here, copy that sequence. There's some very cool other sequences here. We got insulin, very important protein. Here's the green fluorescent protein I was talking about. So this is actually a protein that is similar to proteins that occur in many animals, except it glows green. And so in some situations where an animal is being studied, they'll actually gene edit in this protein so that the structure they're trying to study actually glows inside the animals. Pretty crazy mad science stuff. Collagen, coronavirus spike protein. I'm hoping someone will fold this. The coronavirus spike protein is actually a multiplex protein, though, which means that there's several RNA strands that kind of fold together to create the shape. If anyone does successfully fold the spike protein, though, and you want to try something a little more complicated, this system is capable of folding a multiplex, but it takes a little bit of tweaking. But anyway, so that's some protein sequences that we set up for you guys. If there's any other protein sequence you guys are aware of, would be really happy to help you pull up the sequence to fold it. But then if you just go down to here, there's a section that says input protein sequence. And you can see right there, here's the query sequence. And in order to do it, all you have to do is hit runtime run all. It takes maybe 5 or 10 minutes, but you'll start to see it going through the installation and the processing. And then anyway, go grab the notebook, jump right in. It'll take us 5 or 10 minutes for this runtime to finish. But when it's done, a beautiful 3D image of the protein will pop up right there. And that's just the start. You'll also be able to access all the files. There's lots of pretty cool ways to view and interact with these proteins. For example, mentioned that you can use actually diff-dock or another docking tool to see how a given protein interacts with a given molecule. And also, something I'm excited about is we might try to get some protein VR set up. Because how fun would it be to actually blow up one of these wild proteins to the size of a room and then actually be able to move it around and see its structure and how it works. But anyway, oh, look, it's my filibuster worked, Vivek. I was rambling for long enough that it's starting to finish.
Nice.
But yeah, anyway, and any help needed with the notebook or any questions, like we're open for questions. And anyway, let's fold some proteins, you guys.
For sure. Sounds good. Yeah, thank you guys so much. And yeah, go nuts with the tools. And we're here to help with any of your questions. And yeah, I mean, it's hopefully by looking at this, like you'll see just from these little sequences of characters on a page, these sequences make life possible and are what, make all of us work. So yeah, hopefully by keeping that, the significance of these sequences in mind, you'll be able to gain as much insight as possible out of this. So yeah, have fun with it and let us know if you have any questions. But yeah, thank you guys so much.
Woo! Thank you guys.
Yesterday, German television.
Oh.
At the end, big German channel. We brought them in for you.
Oh, so fun! So glad you're here, and we're gonna be folding proteins for truth and justice today. That's right. I'm actually doing something fun this week, and maybe while we're waiting for this protein to spit out, can tell you guys about it. Because Vivek mentioned something that, ooh, here we go. There's our 3D protein structure. So you can see right there. You can kind of move it around, see what it looks like, and anyone else who folds one who wants to show it, we can get you guys up on the screen too. Maybe we can do a little live protein commentary. Could be fun. But anyway, Vivek shared something which I thought was kind of cool, which is how common this language of genetics and biochemistry is to all life. And one of the reasons why is we all crawled out of the same big old bioreactor, didn't we?
Yeah, absolutely. Yeah, we all crawled out of the ocean from volcanic vents and all of that. So that's why, no matter what microbe you study or any form of life that you study on the planet, the same 4 nucleic acid bases make up their genetic information. It's going to be ATCG for their DNA, and for viruses it's a little bit different. They can be, their primary genetic code can be contained as RNA, the intermediary step between DNA and protein. But by and large, I mean, the same 4 building blocks of our genetic code are present in all life forms on Earth. So yeah, I think that was, to this day, it's one of the most staggering things in science that I've ever learned. So yeah,
it's beautiful and it's remarkable, and I feel like it's one of the reasons why communities like this are so important, is because there is such intersectionality between subjects, and the more we kind of connect with thinkers who use different language and different ideas, I think the stronger our perspectives grow. And anyway, punchline. This week, I'll be working with the world's largest database of marine genomes. So that's genetic data that's floating out around in the ocean. And then what we'll be looking for particularly is enzymes that break down plastic. Because as you are all probably aware, unfortunately, our ocean is completely full of plastic. This is something that's happened incredibly quickly in terms of like the geologic scale. This period of the Earth's geologic history will be looked back on as the plastic era. Yeah. Like we have actually covered the planet with so much plastic that it will be in the strata. It'll be part of geology. They'll be able to date when this mess we've created happened based on the plastic that's everywhere. Yeah. But as you can imagine, when you introduce a new material into such like a biologically complex and evolutionary system, the organisms start to figure out how they're going to use it. So we actually have seen already a number of enzymes evolve in the ocean that can break down plastics. And so we'll actually be using AlphaFold to create structures for all these proteins and then actually do testing to see if they could be something that would break down a plastic. And again, this is just sort of, a new wave of tools and questions that we're able to ask, because of things like AlphaFold. So Very fun. And look, this protein that I put in, it folded quite confidently there, see? So all very high confidence.
Yeah.
Anyone running a protein yet, or?
I have a question.
Go for it.
Stanley, so the input for this protein folding is genomic sequences, right? And then it predicts how the protein folds based on the genomic input. Is that basically how AlphaFold works?
Yeah. So it'll just be the input sequence of amino acids that, of course, come from your genetic information.
Okay. Yeah.
You go from your basic sequence in DNA and RNA of four different types of nucleic acid bases to then to like 20 over 20 amino acid residues,
yeah.
right?
So my follow-up question actually is, so there's a new technology in proteomics called amino acid sequencing from Quantum Sci. I don't know if you've heard of them. Do you think that that'll become the new standard for protein folding, or do you think they'll stick with genomics as the input? Just looking at the, does that make sense to you?
Oh yeah, absolutely. No. I mean, as many computational techniques or approaches to analyzing this overall paradigm in molecular biology, as many of these techniques can be introduced, the better. And I think, that's another important point I think is important for everyone to be aware of is not only do you see, because of the kind of the universal nature of genetic code in all life forms, you can take a discovery or technique, a biological technique from one area or one organism and apply it to so many others, right? In that same way, we wanted to present here, and it's so important for researchers or engineers or scientists and anyone from all different walks of life to get involved with contributing to all this work, is because we never know how a technique developed in one area, like with stable diffusion, will be important if we apply it to protein folding, for example. So,
Anyone know kind of like how stable diffusion works, or what like a diffusion learning process looks like? Because I think this is such a fun one. It's kind of like with diffusion, you start with a structured object and then you destroy it. And then you play back the destruction backwards. So it's kind of like, you imagine breaking a vase and a video of the vase breaking and the pieces flying everywhere. Well, if you played that backwards, you would learn all this stuff about how the pieces fit together. So that's actually how diffusion works in both the art case and the genomics case. It's pretty fascinating, and it's such cool stuff.
Yeah, and actually, as another point to your question, actually, which is, it's helpful to study not only the genomic component, but also specifically in proteomics, because there's a lot of post-translational modification that happens after you go from the genetic to the protein side. So to get a full, accurate, functional picture of what's occurring in the body, you need to analyze both. You need to, or at all stages, you need DNA analysis, RNA, and protein analysis. So to further answer that question, yes, specifically focusing on proteomics is also going to be key. So yeah.
Good stuff, guys. I can answer some questions, or we could start to disperse and work on stuff and hang. But thank you so much for coming and checking us out today.
Yeah, for sure. Thank you guys so much.